Myofibrillar myopathies MFM are a genetically heterogeneous group of neuromuscular disorders, characterized by a common pathological feature: the disintegration of myofibrils, the contractile threads of muscle cells, and the abnormal accumulation of degradation products into protein aggregates. The following is a list of the known genetic subtypes. It's classified by their associated gene and, where applicable, their specific MFM numeric designation. MFM1, desminopathy. This is the most common form of myofibrillar myopathy. Desmin is a key intermediate filament protein that maintains the structure of the sarcomere. It is usually autosomal dominant inheritance. The muscle weakness progresses slowly, that often begins in the distal leg muscles, lower legs, ankles, but can also affect proximal muscles, thighs, hips. Cardiac involvement is frequent and severe, often manifesting as conduction blocks, arrhythmias, or heart failure. It can sometimes precede muscle weakness, and respiratory muscle weakness can also occur. MFM2, alpha-B crystallinopathy. The CRYAB gene encodes a heat shock protein that acts as a chaperone, preventing other proteins from clumping together incorrectly. Symptoms include muscle weakness that is variable. Can include proximal or distal limb weakness. A distinctive feature of this subtype is the development of cataracts, clouding of the eye lens, often posterior polar cataracts, which may appear early in life. Cardiomyopathy, either hypertrophic or dilated cardiomyopathy, is common. Note: a severe recessive form exists, fatal infantile hypertonic MFM, presenting in infancy with stiffness and respiratory failure. MFM3, myotilleinopathy. Myotilleinopathy caused by the myot gene is autosomal dominant. Myotilin is a Z-disc protein involved in sarcomere assembly. Symptoms include muscle weakness, typically later in life after age 50, with distal weakness, most notably foot drop. MFM4, ZASP-related myopathy. Gene LDB3, also known as ZASP, is autosomal dominant inheritance. Encodes the LIM domain binding 3 protein, a structural component of the Z-disc. Symptoms include muscle weakness, usually late onset, 5th to 7th decade, distal muscle weakness, although proximal weakness can occur. Cardiomyopathy. Cardiac involvement, dilated or hypertrophic cardiomyopathy, is a significant feature and can occur in isolation without skeletal muscle weakness. MFM5, filaminopathy. The gene affected is FLNC, filamin C, and is autosomal dominant inheritance. Filamin C is essential for Z-disc stability. Symptoms include muscle weakness. Often presents with limb girdle weakness, proximal muscles of hips and shoulders, rather than the distal weakness seen in other MFMs. Early respiratory insufficiency is a hallmark of this subtype. Cardiomyopathy. Cardiac issues are also common. MFM6, BAG3-related myopathy. The gene affected is BAG3-BCL2-associated athino gene 3. Inheritance is autosomal dominant, however de novo mutations are common. BAG3 is a cochaperone involved in protein degradation, also known as autophagy. Unlike other forms of MFM, this often presents in childhood and is severe and rapidly progressive. Features include severe weakness, rigid spine contractures, and restrictive cardiomyopathy. Neuropathy. Naive axonal neuropathy is a unique pathological feature often seen in this subtype. Genetic advances have expanded the classification to include these rarer forms. MFM7 KY kyphoscoliosis peptidase, a rare, recessive form characterized by childhood onset, generalized muscle weakness, and joint contractures. MFM8 PROX, D1 pyridine nucleotide disulfide oxidoreductase domain 1. An autosomal recessive form that can present in childhood or adulthood with slowly progressive proximal and distal weakness. MFM9 tendinopathy, HMERF. Gene TTN Titin, often associated with hereditary myopathy with early respiratory failure, HMERF. Symptoms: proximal and distal weakness with prominent early onset respiratory failure, often while the patient is still ambulant. MFM10 SV1L supervillain, a recently identified type involving a protein that links actin filaments to membranes. MFM11 UNC45B UNC45 myosin chaperone B, associated with congenital onset, cataracts, and muscle weakness. MFM12 MYL2 myosin light chain to a rare subtype linked to early onset myopathy. MFM13 HSPB8 heat shock protein family B member 8. Previously classified separately as a distal myopathy or CMT type 2L. It presents with distal weakness and hand atrophy. Other associated genes. While not always assigned a strict MFM number, mutations in these genes can produce classic MFM pathology of the production of protein aggregates. FHL1 4.5 limb domains 1 causes X-linked myopathies, including reducing body myopathy. Features include rigid spine, contractures, and muscle hypertrophy. DNA JB6 causes limb girdle muscular dystrophy. D1 LGMD-D1. Biopsies show myofibrillar disintegration and protein aggregation similar to MFM's. PLEC-plectin. Mutations can cause epidermolysis bullosa simplex with muscular dystrophy, EBSMD, combining skin blistering with MFM-like muscle weakness. Thanks for watching.